This is the stomata observed under 10 times magnification. The stomata are open because the leaf was placed in distilled water, which allow water to enter the gut cell. This is the stomata observed under 100 times magnification using distilled water. As we can see, the stomata are open. This happens because in distilled water, the surrounding solution is not concentrated, so water moves into the gut cell by osmosis. The stomata are visible as small openings surrounded by two bean-shaped gut cells. Under the 10 salt solution, the gut cells appear shrunken and less turgid. The stomata pore is either very narrow or completely closed, as the loss of water from the gut cell causes them to become flaccid. When the leaf was placed in 10 salt solution and observed under 100 times magnification, the stomata were seen to be almost closed. This happened because the salt solution made the area outside the cells more concentrated, causing water to move out of the gut cell by osmosis.
The results shown that in flask A, the stalk had no leaves and was placed under normal lab conditions. The dye travelled only about 4 cm. This limited movement occurred because there was minimal transpiration. No leaves mean fewer stomata to drive water upward. In flask B, the stalk had leaves and was also under normal lab conditions. The dye moved up to 5 cm, indicating active transpiration through the stomata in the leaves, which pulled water up the xylem more effectively. Flask C showed the most dramatic result. The stalk had leaves and was exposed to both fan and lamp. The dye travelled approximately 6 cm. The increased airflow and heat accelerated transpiration, causing a stronger pull of water through the xylem.
Now let's discuss some questions regarding this experiment. First, stomata open and close based on water pressure. They open when the gut cell contract and close when flaccid. Second, environmental factors like high temperature, wind and bright light increase water loss because they speed up the rate of transpiration. Next, we don't use latent circulation for water in plants because the water only move in one direction and not return out. The two variables that were intentionally changed were heat and airflow. Flask C had a lamp to provide heat and fan for airflow, both absent in flask B. This change increased the rate of water loss or transpiration, causing flask C much faster to die. Next, to prevent water loss when stressed, a plant primarily closed its stomata. Now let's see how this experiment connected to the lesson. The first of the experiment shows stomata control gas exchange and help the plant save water. The second part shows transpiration moves water through the plant. Factors like heat and airflow increase transpiration, causing the water to move faster.
Lab, I learned that stomata control water loss by opening and closing through gut cells, and xylem transport water continuously, from roots to leaf by transmission pull. I also realized that environmental factors like light, wind and humidity affect how fast plants lose water. By using microscopy, I saw the stomata structure directly, and through the celery dye experiment, I visualized how water moves in the xylem, helping me clearly connect theory to real plant physiology.
Okay, so now let we continue for the reflection on collaboration. Our group observed how stomata reacted in distilled water and salt solution and how that movement showed the rate of transpiration. In distilled water, stomata were open as gut cells were turgid in the salt solution, then closed to water loss. We noticed that the faster the dye moved, the higher the transpiration rate. Our flask leaked, but we worked together to fix it and got better result. This experiment taught we that teamwork and communication are key in scientific work and help us understand